DNA carries the genetic information in the cell. In this presentation, we're going to talk about how a DNA functions. So a gene is a segment of the DNA that codes for the synthesis of a specific protein. Not all regions of the DNA have the code for protein synthesis. Gene is the protein coding segment of the DNA. The essential function of the DNA is to carry the instructions for synthesis of protein. So basically they produce some proteins, and these proteins carry out certain functions in the body. So we said that the DNA carries the code for protein synthesis. So protein synthesis starts with the DNA. And here in this slide, you can see the gene. And gene is a part of the DNA that codes for a protein. Here you will notice there are groups of 3 bases. So here there's one base, and there's another base, and they are groups of 3, and it's called a base triplet. And this is the DNA, and the base triplet will produce, or it will transcribe, a messenger RNA. So this is the messenger RNA, and this is the DNA. So from a DNA, a messenger RNA is created. And in the DNA, you will see a base triplet, that's a group of 3, and a complementary base messenger RNA is formed. And here you can see in the blue, that is the messenger RNA which is formed, and it contains 3 nitrogenous bases. So the guanine is on the DNA, and here it's a cytosine. Where there is an adenine, it is a uracil, and where there is a cytosine, it is another guanine. So this set of 3 is called a codon. So in the gene, you have a base triplet. In the RNA, you have a codon. And the law of complementary pairing is followed. So for every base triplet in the DNA, a complementary codon is formed in the RNA. Also, so here you can see this is a base triplet, and here there's a codon. So that keeps on forming. So the DNA guides or decides the structure of the RNA, and this is called the messenger RNA. So when a codon starts coding, that means it has come across what is known as a start codon. So the start codon begins the amino acid sequence of the protein. That's when the synthesis starts. And also when it reaches the stop codon, that's the signal that indicates the end of protein synthesis. So essentially, this is the process of protein synthesis. There is a DNA. DNA produces a messenger RNA, and it's the messenger RNA which produces protein. So DNA gives instructions to make the messenger RNA. Messenger RNA starts assembling amino acids to make a protein. The step from DNA to production of mRNA is called transcription. And this process occurs in the nucleus. The step from messenger RNA to the production of protein is called translation, and that happens in the cytoplasm. So the messenger RNA leaves the nucleus through the nuclear pores and reaches the cytoplasm. The messenger RNA associates with ribosomes and transfer RNA to produce the protein. So transcription is the process of copying information from the DNA and creating a complementary molecule of mRNA. And transcription happens in the nucleus. Now, there is an enzyme called RNA polymerase, which is the enzyme responsible for transcription. And this is the RNA polymerase enzyme. The RNA polymerase binds to the DNA and opens up the double helix, and it starts reading the bases from one strand of the DNA, and it starts building a complementary strand of RNA. So after transcription, we get an immature form of RNA, and this is called the pre-messenger RNA. And it's a premature version, and the premature messenger RNA contains segments called exons and introns. Exons are the pink segments over there. So you can see this is the exons, and the exons are the segments which codes for proteins, and the introns are the segments that do not code for proteins. So the pink ones are the exons, and the darker colored ones are the introns. So introns must be removed before translation. Introns are removed by a process called splicing. When the introns are removed, the immature mRNA is converted into a mature mRNA. The messenger RNA will leave the nucleus and reach the cytoplasm, and that's where it starts producing proteins. So by a process of transcription, we have created the messenger RNA. Next, we want to create protein, and this process is called translation. There are three main participants in the process of translation: ribosomes, messenger RNA, and transfer RNA. Ribosomes provide the amino acids to build the proteins. The protein is made of amino acids, and the ribosomes, which are located free in the cytoplasm or on the rough endoplasmic reticulum or on the nuclear envelope. These are the three places where you find ribosomes. So the messenger RNA contains codons, which is the 3-base sequence that is complementary to the DNA base triplets. The transfer RNA has got an anti-codon which recognizes the codons on the messenger RNA. The transfer RNA will then go and fetch the correct amino acid and add to the protein chain. So the transfer RNA has got anti-codons which will recognize the codon, and then it goes and selects the correct amino acids. So the assembly of protein occurs in the ribosomes, and this is the process of translation. So here you can see the genetic information is in the DNA, which is in the nucleus. The DNA polymerase has read the DNA, and it has created a mRNA. That's the mRNA which is created, and the mRNA will leave the nucleus and it will reach the cytoplasm. In the cytoplasm, it will bind to the ribosomes, and the tRNA will fetch the amino acids and bring it to the ribosome. The amino acids are added to the protein, and the protein structure expands. When a stop codon is encountered, the protein is released. So here you can see the protein is released. So here you can see the DNA is a double helix, and the double helix is opened up, and the base triplets of the DNA is exposed, and the DNA and the base triplet on the strand of DNA is red, and a corresponding codon of mRNA is transcribed from the base triplet. So this is a base triplet, this is a codon, and you can see there's a start codon which starts the process, and there is a stop codon which stops the process. Then the transfer RNA will come along, and the transfer RNA has got anticodons. So the anticodons of the transfer RNA recognizes the codons of the messenger RNA. The transfer RNA identifies the proteins, the amino acids, and carries the amino acids and links it to the protein chain. So this is a review of the protein formation. The protein structure which is produced is incomplete and is called the primary protein structure. The primary protein structure has to be folded into the correct shape. When folded, the primary protein is converted into the secondary protein, tertiary protein, and quaternary protein. For this, there is another type of protein called the chaperone protein, and the chaperone protein will guide the folding process. For proteins to function, they must have a proper shape. If a protein is incorrectly folded, it is damaged and becomes non-functional. The chaperone protein also helps damaged proteins to be folded back into their correct functional shapes. The ribosomes are found attached to the endoplasmic reticulum, and this gives the endoplasmic reticulum a rough surface, and that's why it's called the rough endoplasmic reticulum. So the ribosomes which are attached to the endoplasmic reticulum are instrumental in protein formation. So proteins are formed in the ribosomes, and they are packaged into transport vesicles. These proteins reach the Golgi complex. The Golgi complex, this is the Golgi complex, and the Golgi complex modifies the protein structure. Here, the protein folding happens, which we discussed earlier. So the protein folding is done, and once the protein folding is completed, it is packaged into Golgi vesicles. The Golgi vesicles will fuse to the cell membrane and expel the contents, the protein, by a process of exocytosis. These proteins will have an effect on cells. For example, insulin is a protein which helps the cells to utilize sugar and lower the blood sugar. So there are many other proteins which are produced, and these proteins have an effect on cells. So the production of proteins by the DNA can be controlled by turning the genes on or off. Cells can turn genes on only when the protein is needed. Cells can also turn off the genes so that protein formation is stopped. Sometimes cells turn off some genes permanently. One example of turning the genes on is production of a protein in breast milk, and this protein is called casein. When the mother is breastfeeding, the mammary glands turn on a gene for producing the protein casein. Casein will be produced and it will be incorporated into breast milk. This is a classic example where the gene turns on and the protein production is implemented.